So this is the last mini lecture for the general biology course sequence. So congratulations for making this far. I assume you're not asleep right now. Maybe it's just sort of playing in the background, and me droning on helps. But the last mini lecture is about genomics, proteomics, and bioinformatics. We're going to talk about viral genomes, bacterial genomes, and eukaryotic genomes, as well as how we study those genomes and what we can get from studying those genomes.
Now, with gene cloning, you can copy genes left and right. Either direction. Actually, it's true because you could copy either side of the DNA if you knew what you were doing. Right. So it helped us really start understanding the genome being the entire set of instructions to build an organism. Now, some genomes are complex, like yours, and some are very simple, like a virus. I want you to understand the difference between the three genomes. First off, we got viral genomes. Like you were saying, viruses contain their entire genome inside this teeny, teeny, tiny head. They're really small. They're a fraction of the size of a cell, like 1/1000th the size of a cell. Yet they have to keep their DNA in there. That means that these are very, very, very small genomes, about 100,000 base pairs or so. Very small. If it was bigger, it wouldn't fit inside of a capsid. And we don't have time. I wish I could tell you. So you guys know about viruses, right? What are the big viruses you know? Humans. The flu. The flu is very variable. You'll notice that every year there's a new strain. Every year the flu adapts. And the reason it's able to is not just mutation, but it's due to what's called alternative splicing. You have one big, long gene sequence, but it may only express two things at once, or three things, or 1 in 3, or 1 in 7, or right. These different combinations trick our immune system because we're ready for one particular type of virus. You are set for last year's flu virus, but it's gone now. There's a whole new virus that's replaced it. So your body has to readapt. So next time you get sick, your body begins to produce antibodies against that particular virus, and then the next year it comes along and it's changed. Moving up, a little bit more complex, are the bacterial chromosomes. Bacterial chromosomes are held within the nucleoid region, because remember, there's no nucleus. And they are inevitably round. Bacterial chromosomes are circles, and they're about a few million base pairs in length. And then you have your eukaryotic chromosomes. It's not just one chromosome, now it's a series of chromosomes in the cell. These are usually huge. They're a gigantic size. Instead of a million base pairs, now we're looking at 10 million, up to 1 trillion base pairs. Bacterial genome versus eukaryotic genome. Somewhere. In order to sequence the genome, we've got to clone bits and pieces of it, and then run these cloned pieces on what's known as a DNA sequencer. We used to have to do it on a bunch of gels and do it all manually by hand, but now we get computers to do it, so there's not really much of a point in talking about how it happens. It is. It takes months. Each little tiny piece. The human genome is sequenced now. We know basically what our manual says, how to build a human. Not how to build a human. We know technically the genetic sequence, but we don't know what every piece does and how they relate to all the other pieces. Of it known. We know the whole sequence, but we don't know what it does. Right. It took 20 sequencing centers about 13, 14 years to get this done. It was expected to take like 40 years.
Do you think about the processing power that would have taken in 1990 compared to like now? Literally do that on the smartphone.
Well, you can't do it on the smartphone. It still does require stronger computer technology, but We have been sequencing things. What took 13 or 14 years then will now take 6 months. That's true. A couple, if you have that many. But the technology has increased so quickly, beyond any doubt. We are leaping ahead as far as technology goes. It's giving us an enormous edge. I just recently, I don't know about you guys, in my house I have this one drawer where all my random electronic cords go. Yeah, well, and it was filled with these cords, and I was getting ready to move, so I was like, I got to get rid of these things. And I had a bunch of old hard drives. And I was like, I don't know, maybe I'll still need these hard drives someday. And then I looked at them, and they were 128 megabytes. That was a lot of data for in the day. And then I was like, there's no way I'm ever going to look at this drive again.
I just bought a 16 gigabyte drive for $7.
There you go. So back in the 1980s, we could clone about 1,000 base pairs per day. In the 2000s, we can clone about 1,000 base pairs per second. So huge difference. Yeah, for a lot less money. Look at that. 2003, it would cost $100 million to sequence the entire genome. Today, it would cost $1,000. Literally, it doesn't take much anymore. And because of that, because it's so quick and it's so cheap, you can have people doing this literally in their backyard labs. No, there's I read a story about this one idiot who swore he was a geneticist. He had a lab in his yard that he made for like $5,000, and he had gotten a hold of some virus material, and I know this is how it all ends, right? He'd gotten a hold of some virus material and some like a hormone sequence, because he decided he was going to make himself stronger. So he injected himself with his homemade concoction of genetic mutation. Yeah, it was a bad idea. The whole goal of this project, the Human Genome Project, was to identify the function of every structural gene and the impact of all the regulatory genes, the activators and the repressors. Figure out how they all work together. We are not there yet. We're getting closer, but we're not there. Because after we start looking at the Human Genome Project, we have to look at the Human Proteome Project. This is identifying all of the proteins expressed in a cell at any given point. Because for every gene you've got, you could have many, 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 many more proteins created. Hundreds of, yeah, we say that now, but 40 years from now, the processing power may be so much higher.
Have you ever read The Last Question by Edgar Allan?
No.
Read that.
Okay. So if our genome is down on this end, the complexity continues to increase almost exponentially as you start to look at the proteome. So it gets a lot more complex. But that's about enough for introductory biology. Now that I've completely scared you away from any sort of future endeavors in bio, the whole point of this is to make people's lives better. You got to remember that as you go forward, if you go into whatever field you go into, do it to be good. Don't be evil. I think that was one of the first lessons we learned at the beginning of the class, right? Don't be evil. Be a good person. Don't be a mad scientist. Try to make the world better. So, do you guys have any questions?
We talked about 3 types of genomes: viral genomes, bacterial genomes, eukaryotic genomes. Size-wise, viral are the smallest, bacterial are the next largest, and eukaryotic are huge comparatively. Viral genomes can be made of DNA or RNA, whereas bacterial and eukaryotic genomes are purely DNA. Bacterial genomes are found usually on a single circular piece of DNA, a single circular chromosome, whereas eukaryotic DNA is found on linear chromosomes. We discussed briefly what genomics is, where scientists can study an organism's complete set of genes. And it's meant to understand how genetic information is passed on and what that genetic information can lead to, what kind of gene products and gene functions it leads to. The proteomics, compared to genomics, centers on studying all of the proteins in the cell at any given point. And again, the goal there is to identify what these molecules are and figure out how they function. So 3 types of genomes, and then we talked about genomics and proteomics. Here are the content review questions to focus your studies. Well, congratulations. That's the end of what I'm going to talk about for the first part of general biology. I do go into the rest of general biology in another class, so keep an eye out for my general biology 2 materials, and I'll see if I can put a link or something into the YouTube comments so that you can find it if you really want to. But otherwise, congratulations, you made it. Now take this information and go be a functional human being and be good to people.